When you need to send a package to someone, you need to pick a good delivery method which deliver your package efficiently, accurately, and safely without damaging your package. Likewise, we are in a search of successful delivery method to deliver chicken NK lysin peptide into chicken gut to mitigate avian coccidiosis. Hello everyone, welcome to my presentation. My name is Samiru Wickramasuriya. I am a postdoctoral fellow at Dr. Hyun Lilloy's laboratory, USDA Agriculture Research Service. Today I am going to talk about Bacillus-based NK lysin peptide delivery system and its effectiveness to mitigate avian coccidiosis in broiler chickens. This is a study that we conducted in our laboratory with the collaboration of US Biology. Before I am going to talk about what we did, I would like to give a general introduction. As we know, coccidiosis is a major enteric disease of chickens that caused by several distinct species of Eimeria protozoa. Based on Eimeria species, they infect different areas of chicken gut. Primarily, coccidiosis damage epithelial integrity in the intestine, and also it decreases nutrient utilization and growth performance of the chickens. According to the literature, it is an annual loss of over $14.5 billion in the poultry industry globally. To control coccidiosis in poultry industry, live attenuated coccidiosis vaccine and some other subtherapeutic medications are commonly used in commercial farms. However, the inability to induce consistent protection, evidence of residual virulence, increasing incidence of drug resistance in Eimeria, and strict government regulations of in-feed medication in poultry production prompted the discovery of alternative strategies to control coccidiosis and to support antibiotic-free meat production. Many scientific studies have been conducted to explore promising alternatives such as probiotic, organic acid, phytogenics, and antimicrobial peptides. In this study, our particular focus was on chicken NK lysin. What is NK lysin? NK lysin is a cationic amphiphilic antimicrobial peptide which identified in natural killer cells of porcine intestine tissue. Later, its characteristic on cytolytic activities were identified against tumor cells and microbes. Chicken NK lysin. Chicken NK lysin, the chicken homologue of human granulysin, is an antimicrobial peptide that showed an antimicrobial activity against Eimeria species via membrane disruption. The characteristics and expression of CNK2 have been previously studied and well documented. Additionally, in our previous studies, CNK2 has been shown to successfully destroy Eimeria species in both in vitro and in vivo conditions. However, effective oral delivery strategy to preserve functional activities of chicken NK lysin in the chicken gut is yet to be discovered. With that, our set objective of this study was to develop a stable strain of probiotic Bacillus subtilis expressing CNK2 to investigate its effectiveness as a carrier of chicken NK lysin to the gut and to explore its protective effect against coccidiosis infection in commercial broiler chickens. Our hypothesis was CNK2 will successfully reach the small intestine and thereby control the Eimeria parasites via membrane disruption. Ultimately, it will improve growth performance as a result of improved gut health together with enhanced immunity. Next, we move to the material and methods. In this experiment, we had 4 treatment groups as unchallenged negative control, Eimeria challenge positive control, Eimeria challenge and treated with Bacillus empty vector group. Finally, Eimeria challenge and treated with Bacillus carrying CNK2 group. We use Ross 708 broiler chickens in this experiment and we received birds on day 1. This is the experimental design. As I mentioned, we received the birds on day 1 and allocate them in a petricime brooder units. And we fed this chicken with diet without any in-feed medications. On day 14, we measure the body weights and randomly allocate into treatment groups. From day 14 to day 18, Bacillus were orally given into the birds in the respective treatment groups. On day 15, Eimeria challenge was performed to all treatment groups except birds in negative control. On 6 dpi, body weight of the birds were measured and set the pan to collect feces. We also collected intestinal tissue samples at this time point to analyze gene expression using quantitative PCR. We stopped fecal collection on 9 dpi and measured the final body weight. All the collected data were subjected to statistical analysis using GLM procedure of one-way ANOVA in SPSS software. Let's move into results. At 6-day post-infection, Eimeria challenge PC and EV treatment shows significantly lower body weight compared to unchallenged control birds. However, birds in NK treatment showed similar body weight compared to the NC treatment. In 9 dpi, body weight trends were similar to 6 dpi data where NK effect was much prominent at this time point. When we look into average daily gain, all Eimeria challenge groups including PC, EV and NK showed lower daily gain compared to birds in NC group. When compare PC and NK showed significantly higher daily gain up to 6 dpi. Interestingly, we did not show any significant difference in weight gain from 6 to 9 dpi among treatment groups. Considering overall period, birds in NC and EV showed similar daily gain and it was significantly higher compared to birds in PC and EV groups. This is the tight junctional gene expression data. As you can see, both NC and NK birds showed similar expression level in JAM2 and sonocludin 1, while lower expression was shown by PC and EV birds. However, birds in all challenge treatments showed lower occludin expression compared to birds in NC group. In mucosal cytokine gene expression profile, both IL-1 beta and IL-2 showed higher gene expression in PC birds compared to other 3 treatments. However, in interferon gamma, all Eimeria challenge birds showed higher expression compared to NC bird. This is what we observed in oocyst data. All Eimeria challenge birds showed significantly higher oocyst shedding compared to the unchallenged bird. Nevertheless, birds treated with NK showed significantly lower oocyst shedding compared to other 2 challenge group. Based on the observed improved growth performance, enhanced gut integrity, and reduced oocyst shedding in the NK group, we can conclude that Bacillus subtilis carrying chicken NK lysin peptide is an effective oral delivery method to Eimeria infected chickens. If you have any questions or interest more on this work, you can read our recent published paper based on our previous study. This is the end of my presentation. I am happy to answer your question. Thank you.